to cover two crash courses, CC number 3 and CC number 4. For CC 3, we will cover amino acids, proteins, and enzymes. And for CC4 class, ang topic natin ay nucleic acids. Now, let's first discuss in brief the content of crash course 3. So here, we will cover amino acid, focusing on their structure and properties. Okay? So for the next topic, ayan, so class, recall in our discussion on carbohydrates, we focused on the glycosidic bond. So parallel ang discussion natin here in a way that we will also tackle the unique bond in your proteins, the peptide bond, and some biologically important peptides. And lastly, of course, your protein structure have levels or hierarchy. So that's our topic for the last subtopic here, the types and levels of structure of your proteins. Okay, so before we formally start our discussion, answer my mini game, yes. So similar lang, 4 picks, 1 word. So here is our item. Sir, ang dali naman, hindi ba yan enzyme lahat? Yes. Ayan. So of course, class, if you're going to ask me, sir, ano po ang relationship ng second picture? So this is actually an assay that contains an enzyme. Okay? So all of these 4 pictures, ang common theme ay enzyme. Ayan, class, get ready for this video. Relatively longer po ang Crash Course 3 than Crash Course 4 because again, we are going to emphasize also enzymes. Alright? So without further ado, let's start with amino acids and proteins. So for carbohydrates, recall, ang building blocks, monosaccharides. For your proteins, ang building blocks natin ay amino acid. And of course, class, I think you are familiar, we have 20 standard amino acid. And these amino acids join together in order to form your proteins, which uses hierarchy in their structure. Okay, so again, class, amino acids are very important in order to carry out essential functions inside our body. So here is the overview of your amino acids. So class, feature, polymeric po ang amino acids and chiral, except for one, okay? Except glycine because of its side chain. Again, polymeric po ang proteins natin, your polypeptides, right? So now, for classification, this is based on the structure because again, class, if you already did an advanced reading in your biochem notes, we have several types of classifying your proteins or amino acids, diba? So here, ang hina-highlight is based on their structural classification, globular and fibrous. But of course, we can also categorize them based on their functionality and solubility. So for the specific and unique bond type, ayan, si peptide bond, which in nature is amide, diba? Amide functional group. So I do hope that you still remember the amide functional group. And lastly, for the key function, ayan, main function ni proteins, particularly a specific type of your protein is for catalysis. So aside from that, it can also participate in signaling as well as in structural support. Clear ba, class? Alright, so now let's proceed with the structure of your amino acids. So although 20 sila, they have a general structural feature. Ito ang structural feature ng amino acids. So let's dissect each atoms here. So this carbon at the center is called your chiral carbon because again, it has 4 different functional groups attached to it. Alright, again, except glycine. So as you can see here, at the left side, it has an alpha amino group, and at the right side, it has a carboxylic acid group. And both of them, class, remember, they are ionizable. Sir, what do you mean ionizable? Meaning they can be protonated or deprotonated. So the third group is just a hydrogen atom. Sir, ano yung pang-apat? So that's actually this fourth functional group gives your amino acids their unique characteristic and property, and that's your R group or their unique side chain. Okay, so to repeat the general structure of your amino acid, we have chiral carbon bonded to alpha amino, alpha carboxylic acid, hydrogen, and a unique side chain. All right, so here is the structure of your 20 standard amino acids. So class, remember ha, meron kasi tayong non-standard amino acids or technically derivative po siya ng 20 standard amino acids. For our discussion, we are just going to focus with the 20 standard AAs or amino acids. All right, so no need to familiarize the structure. Class, ang important here is that you know their groupings depending on their side chain. So for this table, we have 5 different groups. Okay, so first one, this is the non-polar group or those amino acids that only contains alkyl group in their side chain. So we have alanine, valine, leucine, and isoleucine. Actually, guys, si glycine and proline, they are also in this group. All right, they are relatively non-polar, but of course, because of their structure, unique sila. Okay, that's why we will give emphasis to those two, kaya wala sila sa table. All right, next grouping, those that have hydroxyl or sulfur atom in their side chain. So again, we have 4, serine, may hydroxyl group, threonine, merong hydroxyl group. Si cysteine and methionine, sila yung may sulfur atom. Particularly class, si cysteine, it has a thiol functional group in its side chain. And this is very important in creating disulfide bonds, stabilizing the structure of your proteins. Ayan, very unique po si cysteine. For carboxyl group, we have 4 as well. Aspartic, glutamic acid. Sir, meaning meron silang carboxylic functional group? Yes, very obvious on their side chain. Si asparagine and glutamine are just like your aspartic and glutamic acid respectively. O anong difference? Instead of your hydroxyl or your carboxylic, sa side chain nila, they have an amide functional group. Alright? So later on, we will see also that the aspartic and glutamic acid are example of acidic amino acid. So meaning meron silang extra ionizable groups sa side chain nila. Because very important yan for our calculations. Ay sir, may calculations pala sa biochem. Well, relatively easier naman ang computations natin here. Alright, so going back, for basic amino acids, we have 3. Si histidine, lysine, and arginine. For lysine and arginine, they have an additional ionizable group. As you can see here, diba? They have an amino group, which is very basic. Okay, class, again recall, kapag amino, basic. Kapag carboxylic acid, acidic. Alright, and then lastly, we have the aromatic amino acids, si phenyltryptyr. So that's how I familiarize them. And class, as you can see, they all have aromatics in their side chain. And because of that, these are the only amino acids capable of UV absorption. Or remember, very unique ang aromatic amino acids because again, they are the only capable amino acids of UV absorption. Okay, so now let's proceed with some classifications of your amino acid. So similar with fatty acids class, we can classify your amino acids with 2 classifications or broad categories. We have essential and non-essential. Similar lang with FAs, pag essential, we need or it must come from our diet because we cannot synthesize it readily. Although, meron namang instances that our body is capable of synthesizing these, however, it is not a priority. O sir, bakit? But may prioritization because it is energetically unfavorable. Okay? So in order to familiarize your amino acids, ayan, we have a mnemonic. O sir, both ba yan need namin i-memorize? So class, ang tip ko here is just memorize the essential amino acids. Okay? And then the rest, of course, non-essential siya. For essential, we have this mnemonics. Try this VIP mol. Now, class, careful lang ha, because ang amino acids nyo, diba, as we all know, they have one letter and three letter codes. Yung mnemonics natin here, it does not, okay? It does not follow the official one letter code. Okay? It's just the initials of your amino acids. So for try this VIP mol, T stands for tryptophan, T stands for threonine, his or H stands for histidine, V valine, I isoleucine, P phenylalanine, M methionine, and for 2 L's, lysine and leucine. And then the rest, of course, non-essential. All right, class, actually we have two unique amino acids in terms of their grouping. Si histidine, this is also considered as semi-essential. So for infants, because they cannot synthesize it, essential siya. But upon reaching adulthood, somehow we are capable of synthesizing this through a specific pathway, which we will discuss on amino acid metabolism. And that's similar with your arginine. Semi-essential lang po siya. All right, so now that we already introduced to you the 20 standard amino acid, ayan, quick quiz muna tayo on general info about their unique properties. O sir, bakit? Because sometimes, class, this appears in your board exam. Again, trivia heavy on items sa board exam. So it's better that we feed you those that are known to appear. Okay? Appear from the past board exam until the last year's board exam. Alright, so number 1, the only amino acid with R configuration. So meaning ba yan, sir, lahat ng amino acid as ang configuration? Yes po. Only one is in R configuration, and that is your cysteine. It's because of the sulfur atom. Mas pinaprioritize kasi ang sulfur because of its higher atomic number. O, diba, recall your organic chem in assigning R versus S configuration. Alright, number 2, the only achiral amino acid. Alam na natin to, that's your glycine, diba? Number 3, the only secondary amino acid. So meaning, class, your amino group is attached to 2 carbon atoms. The carbon of your amino group is directly attached to 2 carbon atoms. That's your protein. All right? So here is the structure kasi of protein. It's bulky because of the secondary amino group here. Diba? As you can see, ayan, it is bound or bonded to 2 different carbon atoms. Number 4, has an indole side chain. Familiar with the indole side chain or indole ring? So this is actually a characteristic of tryptophan. So class, yung ring structure here, the 2 cyclic rings here are your indole group. All right? And it's largest by molecular weight. All right, last 2. Acts as a buffer near physiological pH because of its imidazole side chain. O sir, parang hindi ulit ako familiar. O class, remember, the structure of your imidazole is very unique for histidine. And also, it acts as a buffer because, again, ionizable din ang amino group ni histidine. Alright, so last one, it has a guanidinium group and it's highly basic. Oh, class, mamili na lang kayo dun sa 3 basic amino acids, which is which, diba? Alin ang may guanidinium group? That's your arginine. O sir, alin din yung guanidinium group? So that's the structure at the end. Those that contains nitrogen atom and the carbon atom doubly bonded to an amino group. Alright, so I hope that's clear. Class, please take some time in order to memorize this. For biochem in general, my tip is, so yung study habit kasi na ginagamit ko last during my board exam preparation is through flashcards, either digital or physical. Okay? Very efficient siya in order to remember very crucial information. Alright? So again, class, flashcards are very important, particularly here in biochem. So although very information heavy tayo, wala namang computations at all. Or very algebraic lang yung computations. Alright? So class, as I have said earlier, may ionizable group ang amino acids natin. That's why it can be affected by pH of the environment it is situated in. Diba? So that's why meron din siyang characteristic pH. Familiar with the term of the characteristic pH ng amino acids. That is your isoelectric pH or isoelectric point or PI ang abbreviation, which is the pH of your molecule in which your amino acid has no net electric charge. So it does not mean, class, na wala na siyang charge at all. No net charge lang. So as you can see here, this is the example of our Zwitter ionic form. So we call your amino acids with no net electrical charge as Zwitter ionic. Okay? So here, ang amino group, protonated. Ang carboxylic acid group, deprotonated. Hence, ang kanyang net charge is 0. So it's in its isoelectric pH. O sir, may computation ba? Yes. So we have a formula. Very easy lang, class. Don't be scared. Ayan. So here are the 3 different computations. But again, class, similar lang yun. You will only use different pKa for computation ng acidic and basic amino acid. So basically, class, to conclude, ang computation here is that summation ng 2 pKa's divided by 2 is the PI or the isoelectric point of your amino acid. Okay? So now let's take example for each type of amino acid. Ayan. So first, glutamic, glycine, and lastly, lysine. Glutamic acid, as you can see, it has an extra ionizable group. Glycine, neutral siya. Lysine, basic. So of course, meron siyang additional ionizable group. And class, just a tip, if you're confused, which values should you use to plug in the formula? So first one, if an amino acid side chain contains an acidic or basic group, or basically they also have, or they have 3 pKa values, always remember na ang gagamitin yung pKa values are those that are near to each other or close to each other. Okay? So for instance, sa glutamic acid, you will use those pKa's near to each other. So that's your 2.2 and the 4.3 or the pKa of the R group. Alright? For instance, for lysine, basic. So again, 2 near pKa. So that's 9 and 10.5. Alright? So that's just a tip. So you can use it as well in order to guide you in computing the pKa of acidic and basic amino acids. So class, again, that's only applicable for acidic and basic amino acids because they are the only amino acids that have additional pKa. Alright? So let's see the titration curve of those 3 examples. Ayan, starting with lysine. So here are the 3 structures of lysine depending on the pH of the solution. Okay? So here is the titration curve. Now, this structure of your glycine exists when there is a low pH. Again, low pH to class because protonated pa both si amino and carboxylic acid. So below the pKa or PI ang structure na to. Alright? So as you can see here, ang net charge na is positive 1. Now, as we increase the pH of the solution wherein it is equal na sa PI, ayan, Zwitter ionic na si glycine. May protonation sa amino group but deprotonated na ang carboxylic acid. Because again, class, relatively higher pH na po siya, meaning more basic than the previous pH of the solution. Alright, and then lastly, ayan, as you can see, fully deprotonated na si glycine because it already reached beyond the pKa of your amino group. That's why deprotonated na si amino group. And check, ayan, -1 ang net charge ni glycine. Alright, so now let's take your glutamic acid. So again, class, glutamic acid is an acidic amino acid. So here are the structures of your glutamic acid. So compared with glycine, na meron lang 3 structures or 3 predominant structures, si glutamic acid, because of the additional ionizable groups, it has 4 structures. And here's the titration curve of your glutamic acid and its sweeter ionic nature. So again, class, PI niya is around 3.25. So ang baba, because again, glutamic acid is an acid because of the carboxylic acid and its side chain. Ayan. So of course, for low pHs, lahat po ng ionizable group are protonated. Okay? Very simple na lang yan, class. At very low pH wherein hindi pa na-reach ang pKa or pH is less than your pKa, protonated lahat ng ionizable groups. Okay? Upon reaching pK1 or pH is equal to pK1, ayan, it starts to give up its hydrogen atom. Deprotonated na si carboxylic acid na naka-directly attached to your alpha carbon. Class, remember ha, laging unang nade-deprotonate ang carboxylic acid attached to your alpha carbon. Okay? So upon reaching your PI, 0 na po ang what do you call this, net charge ng glutamic acid. And this is the structure of it. Okay? So now, upon reaching your pK2, ayan, so ito na siya, class. When you reach your pK2, pati yung ionizable group in the side chain is already deprotonated. So this is the region in the titration curve. Alright, and then lastly, upon reaching pK3, wherein deprotonated na rin si amino group, your glutamate or glutamic acid has a charge or net charge of -2. Okay? So ang trend here, guys, for your neutral amino acid, ang net charge na lang ay from +1 to -1. Okay? So for your acidic amino acid, from +1 to -2. Alright? And you will see later on sa basic amino acid, so I will not discuss this comprehensively. So similar lang po siya sa acidic amino acid. But here ang difference, ang net charge is from +2 to -1. Okay, class, similar lang yan. Ang unang madedeprotonate always ay yung alpha amino group. And of course, as you can see in its titration curve, sir, ilan po ulit ang pKa? 3 po, or 3 pKa values. Alright? Now, here is the PI of your lysine amino acid, or relatively higher than your glutamic acid. Because again, in nature, basic C amino acid, lysine. Ayan, to summarize our discussion, here is the table that summarizes the relationship of your pH and pKa and the net charge. So class, ito yung summary ng discussion natin for the 3 examples. Alright? So please take some time to review this summary. So we will give you a copy of the slides. Ang main takeaway lang here, again, is that, of course, kapag pH is less than pKa or in acidic environment, net charge is always +1 for neutral amino acid. And of course, fully protonated ang structure. Lahat ng ionizable groups have a hydrogen atom. Okay? So for isoelectric point, deprotonated si alpha carboxylic acid, but protonated pa po si alpha amino group. That's why net charge 0, we call the structure Zwitter ionic. And lastly, for your last relationship, kapag highly basic na or your pH is greater than your pKa, lahat po ay deprotonated na, and then ang net charge is -1 or -2. It varies depending on the nature of your amino acid. Fully deprotonated. Alright? So that's it for isoelectric pH. We are done with the structure and properties of amino acids. So similar with carbohydrates, class, transition discussion natin ang unique bond that joins your amino acids together. Okay, so for this visualization, we have 2 amino acids. So similar reaction mechanism. Upon dehydration, wherein we liberate water, we will form an amide bond. So class, disregard the extra O here. So ano lang to? Mistake lang in drawing the structure. So again, amide bond is CONH. Okay, so here, polypeptides are written using their 3 or 1-letter codes. So ang example ng 3-letter code, we have glycine, alanine, and serine. Ang directionality, class, ayan, N-terminal to C-terminal. So meaning N-terminal is the amino acid at the left side. Alright? And then the C-terminal is the last amino acid or the amino acid at the right side. Alright? So ayan, class, ang formation ng peptide bond. Sir, parang mas madali siya compared sa glycosidic bond formation. Yes po, di ba? Because sa glycosidic bond, we have anomeric configurations. Here alpha and beta. Here very straightforward. Okay, class. So here is this is just a visualization of your formation ng peptide bond. As you can see here, we liberate your water molecule in order to form a peptide or an amide bond. Okay, so now let's proceed with the discussion of your peptides, knowing that the bond that links these peptides together are your peptide bonds. Okay, so these are biologically important peptides. For this discussion, class, we are going to focus on peptide and protein hormones. Or remember, class, ang hormones, we will encounter it more on our discussion of metabolic pathways. Ang hormones kasi, they participate heavily or greatly in enzyme regulation. Okay? So second one, we also have neuropeptides, peptide antibiotics, and toxins are used in medicinal and clinical chemistry. Okay? So for this discussion, again, ang ating focus are your peptides or biologically important peptides and protein hormones. Okay? So here is an example of your protein hormones. Familiar with glucagon and insulin? O diba? Sila ang main hormones that regulate carbohydrate metabolism or actually metabolism or metabolic pathways in general. Okay? So yung discussion for this two will be in detail sa regulation part ng metabolic pathways. I hope that's clear, class. So now, let's proceed with our, ayan, ang favorite nyo, quick quiz. So first item, it regulates blood sugar levels by facilitating the uptake of glucose into cells. O class, nakita nyo na siya earlier. So again, ang root word here, uptake of glucose. So this is your insulin. O diba? Next, raises blood sugar levels by promoting breakdown of glycogen. O sir, ano po ang clue? Clue is raises blood sugar. O diba? So of course, you will encounter this more again sa carbs metabolism. That is your glucagon. Or ang mnemonic ko dyan or ang tip ko is it raises blood sugar level because it signifies glucose is gone or glucagon. O diba? So third one, regulates social bonding, reproduction, and childbirth by stimulating contractions during labor and milk ejection during breastfeeding. Ito class, very my favorite hormone, di ba? So that's your oxytocin. Okay? And last, I'm sorry, second to the last, this regulates water retention in the body and blood pressure. So this one, it's not commonly known. So it starts with letter V. May hint na kayo class. This is your vasopressin. Okay, so class, please take note this, biologically important peptides. And lastly, this regulates appetite and metabolism by signaling to the brain when the body has had enough food. So parang hirap naman yung last two. So yung last peptide class is starting with letter L. So actually class, I'm not familiar with this as well during my review with the board exam. So our answer is leptin. Okay, so please remember these 5 biologically important peptides as these are very known to appear in the board exam. Okay, so we are done with the peptide bonds and the biologically important peptides. Now, we will discuss the levels of protein structure and its classification. Okay class, carry pa ba? So class, again, you can take breaks if ever na saturating ang discussion natin. That's really the nature of your biochem subject. Okay, so again, take sanity breaks every now and then. So in my on-site class, I give them 5 to 10 minutes before I proceed with the next crash course. All right, but now let's proceed with our next topic, proteins, levels of structure through this video. So I will play the video class and then we will rationalize what is the main takeaway of this video.
The shape of proteins. There are 4 different levels of protein structure or protein shape: primary, secondary, tertiary, or the 3rd level, and quaternary, or the 4th level. The primary or 1st level of protein structure is a chain-like or linear sequence of amino acids joined by peptide bonds, as you can see here. The secondary or 2nd level has 2 shapes: an alpha helix or coil-like formation, or a folded pattern called a beta sheet. In both of these structures, hydrogen bonds between the amino acids stabilize the shape of the protein. The tertiary or 3rd level of protein structure involves more folding and bonding of the secondary structure. The coils may even touch each other, as some covalent bonds form from the sharing of electrons between different amino acids, but most of these twists and folds occur from the result of ionic bonds between positively and negatively charged R groups of the amino acids. The quaternary or 4th level is where clusters of more than one polypeptide chain link together to form a giant molecule. Many different types of bonds may be formed within this structure. The shape of protein is important. For instance, fibrous proteins are extended linear proteins that are part of ligaments, tendons, and muscles. And globular proteins fold into almost spherical shapes and have their hydrophobic or water-fearing R groups buried deep within the core, and their hydrophilic or water-loving R groups extended out into the water when in an aqueous environment. Proteins take their shape based on the job they're required to perform, and some have moving parts that are important to their functions. Proteins can have their structure or shape altered by denaturation. When this happens, the protein is no longer able to carry out its job. Denaturation can take place because of change of temperature, change in pH, or the presence of certain hazardous chemicals. And that be the basics on the shape of proteins.
Alright, so guys, from the video, we have seen that really your proteins have 4 levels of structure. Diba? Very comprehensive yung video. It already tackled the classification, the structural classification of your proteins, diba? Your fibrous and globular. But for this, ang main takeaway natin is that its structure is composed of 4 different levels. So we have primary, secondary, tertiary, and quaternary. So starting with primary, so actually class, this is just the sequence or the building block sequence of your amino acids. And of course, the bond that joins these amino acids are your peptide bonds. So this is an example of the primary structure of your proteins. Okay, so this comprise a peptide or a polypeptide chain. So as you can see class, linear lang siya. So we linearized it in order to easily visualize the primary structure. Okay, so now let's proceed with the secondary structure. Or recall from the video, it stated that we have 2 main classifications of secondary structure. We have alpha helix or beta pleated sheets. So class, the result of the folding is due to hydrogen bonding between the amino group and the carboxylic acid group of non-adjacent amino acids. So hindi necessary na nearby yung amino acids because again, your proteins will fold eventually. And the stabilizing interaction here in your secondary structure is hydrogen bonding. So parang I recall, during our undergrad, we also discussed super secondary motifs and domains. Do we need to study it here for boards prep? So actually guys, for the past board exam, they do not include those advanced concepts, okay? That's why our focus here is just for these 2 secondary structures, the alpha helices and si beta pleated sheets. So ito class ang structure and visualization ng alpha helices. As you can see, non-adjacent amino acids ang nagkakaroon ng interaction through hydrogen bonding. For beta pleated sheets, the same is true. As you can see, here is the example of hydrogen bonding. Okay, so let's proceed with another highlight of our secondary structure. O di ba kanina I said that very unique or very special si glycine and proline because they disrupt the formation of alpha helices. Ay, true ba sir? Anong reason? So of course, for glycine, here is its structure. So what do you think, class, is the reason why it disrupt your alpha helices? So class, the reason behind is because it's relatively small. That's why it gives many possible angles of rotation. And we do not want that on your alpha helices as it destabilizes the structure of your secondary structure. Of our proline naman, here is the structure. So the opposite is true. Kung si glycine is very small, si proline, due to its side chain, the secondary amino group is bulky. Hence, it limits its flexibility. O, diba? So glycine and proline disrupt alpha helices for opposite reasons. Okay, so now class, I prepared a protein in focus. So meaning, an example of a protein. Can you guess what protein is this? Sa mga skincare enthusiast, you can guess. So this is actually your collagen. Very different ang secondary structure ng collagen because it's not an alpha helix. Instead, it is composed of triple helix and that's why very extended po ang linear structure ng collagen. And of course, this gives your collagen the elasticity and strength for structural support. So as you can see class, it has a repeating pattern of amino acid. So very common yan kapag ang role ng protein nyo is for structural support. So for collagen, it has a repeating gly, XY, where X and Y are your proline and hydroxyproline respectively. So this is the hierarchy of organization for your collagen. So these are the 3 main amino acid. And then of course, it forms your triple helix and eventually lastly, it will be tightly packed to form your collagen fibers. All right, that's why very elastic, or it gives elasticity, diba, for the structure of several organisms. Now, we proceed with the tertiary and quaternary. Class, I forgot to emphasize pala earlier that not all proteins have quaternary structure. Okay? Enough yung tertiary structure lang for them to be able to functionally active. Okay? Again, not all proteins that are functionally active have quaternary structure. Alright, so going back, for tertiary, this is just the 3D shape of your polypeptide. As you can see, meron din tayong mga stabilizing interactions for your tertiary structure. First one, we have the interactions of your side chains, particularly through hydrogen bonding. So similar with secondary structure, disulfide bridges. Oh sir, di ba yan yung cause? I mean, yan yung effect ng cysteine sa amino acid chain? Yes. Okay, and then of course we also have weak interactions through electrostatic and hydrophobic interactions. Okay, so lastly, quaternary, this is just the interchain assembly of your protein subunits. So sir, yung subunits ba, tertiary structure po yan? Yes po. Okay, so here for your example of protein, this is composed of two different tertiary structure assembled in order to form your quaternary structure. Again guys, hindi required or hindi siya prerequisite your quaternary structure for your proteins to be functionally active. Okay, so that's it for protein levels of structure. Again guys, for your board exam prep, hindi ganun ka-complicated ang need nyong malaman or need nyong ma-familiarize for the levels of protein structure. Okay, so now we proceed with the structural classification. O, diba? I emphasized this earlier. We have two, fibrous and globular. Okay, let's discuss that in detail. So these are the structures of fibrous and globular proteins respectively. As you can see in their shape, si fibrous, long and narrow, and it's very important for its main purpose of structural support. Okay, si globular, usually class, it is spherical, but it still varies in terms of its shape. Sa purpose naman niya, it has a diverse functionalities depending on the genes that code this globular protein. Okay, so for amino acid sequence, oh, diba? I emphasize this for collagen, for instance, because structural siya, repetitive ang sequence ng amino acid. The same is true for your fibrous proteins, diba? So because it's for structural support, repetitive din ang sequence ng amino acid. On the other hand, si globular, irregular ang amino acid sequence because its functionality or purpose is dynamic. All right. For durability, we will tackle this more on the part of enzymes because again, proteins in general are sensitive on fluctuations in their environment such as in temperature, pH, and extreme conditions. All right. So for examples, no need to memorize this. So if in our discussion class, you will see that there are certain overlaps with the examples of your different classifications. Okay, and lastly, for solubility, class, remember our discussion on carbohydrates. Kapag tightly packed ang structure ng biomolecule or ng macromolecule nyo, it is insoluble in water. Okay, that's why si fibrous protein insoluble, while si globular is generally soluble in water. Okay, they are capable of hydrogen bonding. Now we proceed with our second classification. All right? So ito class ang ating second classification. We have four. Sir, lumalabas yun sa board exam? Yes po. There is a certain classification here that appeared, okay, for 2 consecutive years starting from 2023 and chika ng student last year din nag-appear ulit siya. So that means very important that you familiarize this. Okay? So I will emphasize this on our discussion. So first one, albumins. Class, I will just focus on the solubility of this classification. Okay? Si albumins, water soluble. Okay? Ang common example, egg albumin. O, diba? Si globulins, of course, they are soluble in saline or dilute salt solutions. For instance, we have immunoglobulin. So ito class ang visualization or picture ng immunoglobulin. I think this is immunoglobulin A. For prolamins, ayan, sa alcohol, particularly class 60 to 80% ethanol lang siya soluble. O sir, ito ba yung lumabas? Because you're very particular. Yes, si prolamins po ang lumabas sa board exam. So same na property, soluble in 60% to 80% ethanol. O class, please remember ha, prolamins again, ethanol, soluble or alcohol in general. So examples, we have zein, component of corn, and gliadin, a component in wheat. Okay? So lastly, we have glutalins. So si glutalins, acid and base, soluble po siya. Alright, so let me repeat. Albumin, water soluble. Si globulin, it's saline soluble. Prolamins, alcohol soluble. Glutalins, acid base soluble. Sir, baka naman may panemonics ka dyan. Yes po. So remember, SPAG. Mga favorite, mga pasta lover, SPAG means solubility for prolamins, albumins, globulins, and glutalins. Alright, so now we arrive with our last classifications. So functional. So actually class, we always include this in our discussion. Very important to in order to transition to our next topic. Because again, majority of your proteins, they have enzymatic roles. The first classification here in their functional category. Okay? So here, very straightforward. Enzymatic meaning for catalysis, transport for transport or membrane transport of molecules, structural, storage, and signaling. Okay, class, very straightforward yung properties ha. So I'm not going to discuss this in detail. So you can read this in your manual. For the mnemonics, familiar sa game na Tetris? Ayan. Tetris means functional, which is enzymatic, transport, structural, storage, and signaling. Okay, class, I hope it's clear. Again, we have three classifications. First one is structural, second is solubility, and third is functional. Now is my final last words for proteins and amino acids. So class, brief recap lang of what we have discussed. So this is our ayan, so amino acids and proteins. For our summary of monomer, amino acids again ang building block. Class, I emphasize the directionality and terminal to C-terminal. Class, naturally occurring forms of your amino acids is in L configuration. So relative to your carbohydrates, di po ang sa carbohydrates. So D-carbs, L amino acids, or ang tipo dyan, lamino acids. Diba? So energy function, yes, but only during starvation. So mas priority po natin ang carbohydrates and lipids as our source of energy. So for digestion mechanism, we have your proteases as the main enzyme and site of digestion, stomach, and small intestine. And solubility, lastly, depending on classification, it is variable. Alright, class, again, before I forgot, sa biochem notes nyo, we already compiled the summary of your biomolecules. I think it's after the discussion of nucleic acids. Okay, so going back, we also discussed the levels of protein structure. For primary, ang hint natin dyan, amino acid sequence. Secondary, local 3D arrangement. For instance, alpha helices and beta plated sheets. Sir, ano nga po ulit yung stabilizing interaction? Hydrogen bonding. Very good. So for our tertiary structure, that's your 3D shape of a polypeptide. Alright, so for quaternary, we have the assembly of protein subunits. And lastly, class, please remember our 3 classifications. For structural, we have fibrous and globular. For solubility, we have 4. Class, focus on prolamines, again, ethanol, 60 to 80% in particular, soluble. Okay, albumines, globulines, and glutamines are also the classifications in terms of solubility. And lastly, we have your functional classification. And this is the summary of our discussion on amino acids and proteins. Okay, class, before we proceed with our next discussion, take a break muna. All right, so medyo saturating na at this point, right? So for our next discussion, we will tackle enzymes. Okay, similar lang naman yung format. We have your structure or its components, classifications, and some special topics. Okay, so for now, class, I will proceed now with our next set of discussion, your enzymes. Here, we are going to first prioritize the composition and classification, and then the mechanism and models of substrate binding. And for the last 2 topics, this is more on the kinetics, inhibition, and regulation of your enzyme. Now, class, remember ha, sa enzymes, they both have a catalytic and regulatory functions. Okay, so again, here we will prioritize your components of enzymes. So again, ang main function po ni enzymes is for catalysis. This is the component of your enzymes. First, we have your cofactor and then coenzyme. We also have your catalytic site. Same, same ba yan with your active site? Yes. All right. So we also have your apo and holo enzyme. So now, class, let's put our focus here on their description. Si cofactor, that's your non-protein molecules. It can either be organic or inorganic. Typical cofactor po ng enzymes natin, as you will observe on our next discussions, are your metals. Okay, for instance, magnesium 2+, they serve as cofactor in order for your enzyme to be functionally active. Next one, we have your coenzyme. These are your organic cofactors. So meaning class, si cofactor is an umbrella term because si coenzyme is very specific, organic lang siya. So recall our B complexes, those are examples of your coenzymes. All right? For catalytic site, again, this is your substrate binding site. And then for apoenzyme, this is the inactive part of your enzyme, the protein portion of the whole enzyme complex. And lastly, once we assembled all of the needed components, we call your whole enzyme as holoenzyme. Kaya nga diba holo enzyme, which is the active form of the enzyme. Class, clear ba with the components of your enzymes? All right, so now we will proceed with enzyme classification. Of course, class, very diverse kasi ang enzymes. That's why we have a very, very long list of enzymes. So of course, biochemists, they agreed to have a system in classifying and identifying enzymes by the reactions they catalyze. So ito class, this table is just a supplementary discussion. All right. So for instance, we have your enzyme ascorbate oxidase. So its enzyme commission number or identifier is 1.10.3.3. And each number here corresponds something. O, diba? So you can see here, EC1 meaning it's an oxidoreductase enzyme. All right. So no need to memorize the subclass, the subclass, and the serial number. Again, ang takeaway lang natin here is that it's very important that we have a system of classifying your enzyme. Alright, so here we are going to focus our attention to the first classification or the enzyme commission classifications. So first class, quick quiz muna ulit tayo. We have redox reactions involving substrates. So ang galing naman yan, nasa description na, that's your oxidoreductase, your EC1. O class ha, please be cautious, bawal mag-switch ang enzyme classification. They are very particular. Si EC1 ay oxidoreductases. Next, we have this transfers functional groups between 2 substrates. So, sir, nandun na naman sa description. Transfer, di ba? So, transfer races. Alright, so that's your EC2. Very easy. Number 3, we have your hydrolysis by the addition of water. Again, class, ang clue natin dyan is hydrolysis. That's your hydrolysis enzyme or your EC3. Okay, so fourth, we have the addition of a group to a double bond or elimination of a group to create a double bond without hydrolysis. So actually, class, this is the opposite of your hydrolysis. Ang ating clue here is the term without hydrolysis. So that is your, any guesses, lyases, di ba? Number 5, transfer of groups within substrates to yield isomeric forms. O class, ang hint natin dyan is the term isomeric. From that term, we know that these enzymes are your isomerases, the EC5 enzymes. Okay, second to the last, formation of bonds by condensation reactions coupled to ATP cleavage or similar cofactor. So meaning it uses energy via ATP. That's our clue. Here, that's your ligases. And lastly, class, relatively new classification, movement of ions or molecules, across membranes or their separation within membranes. And that is your translocases. So actually, guys, dati, this classification is under transferases. But again, very unique kasi ang function ng translocases in a way that they transport molecules in membrane-bound organelles or of course, in transporting from one organelle to another. Okay, so to summarize this classification, and of course, class, again, as we have said, very important that you are familiar with vitamin-related coenzymes. So, sir, do we need to memorize this? Yes po. Alright, class, check your biochem notes at the last page. At the very last page, we summarize the list of abbreviations. So, if you're not familiar with this vitamin-related coenzymes, you can check their whole meaning in the list of abbreviations. Okay, so, sir, meron kang mnemonics for enzyme classes? Yes po. Okay, so feel free to use our mnemonics. Here is, sa mga alcoholic, o diba? O tagay, wag lang isang litro te. We have oxidoreductases, transferases, hydrolases, lyases, isomerases. O check guys ha, litro Li, ligases. Very important because we have 2 L's in our mnemonics. And lastly, translocases. Alright, so that's it for our classification. Now class, I prepared again a quick quiz. So these are your main digestive enzymes. So we already tackled the 2 types of digestive enzymes, particularly for your carbs and lipids. Now, you will determine these enzymes or these 4 enzymes that are our main digestive enzymes. Okay, so ready na class? So first one, you will find me in saliva. Second one, I break starch down into maltose. Ay, sir, alam ko na yan. Alright, so wait lang class, we have the third hint. I'm often the first enzyme to start digestion. Alright, class, our answer is amylase. Very good. So for our second digestive enzyme, I'm released by the pancreas. Medyo vague pa because many enzymes ang nire-release ni pancreas. This is the second hint or second clue. I need bile salts to do my job better. O sir, parang familiar si bile. Parang na-discuss mo na yan earlier. Yes po. So this is the third clue. I break down fats into glycerol and fatty acids. Okay, class. So I think you already know the answer. Our answer here is lipase. Alright, so for our third digestive enzyme, ayan, I'm found in your stomach and pancreas. O sir, pwede pang next clue. My specialty, cutting peptide bonds. Ay sir, parang alam ko na. Alright, ang clue natin, peptide bonds, so meaning for proteins siya class. O, diba? I help break big proteins into smaller pieces. So class, ang general term natin for the enzyme that degrades or process your proteins are your protease. Alright, and then lastly, I think, ayan, alam nyo na, this is for your nucleic acids. Let's check the clues. First, I snip phosphodiester bonds like scissor. Second, I target DNA and RNA. O, diba? And third, you'll find me in cells cleaning up nucleic acids. And answer, nuclease. All right, good job, class. So now, actually, our focus here is just protease because again, this is for the discussion of proteins. So formally defined as breaking peptide bonds to digest proteins into shorter fragments. Okay, so protease class, we can further classify them into two categories. The first one, ayan, it can only act upon terminal amino acids peptide bonds. Okay, we call them your exopeptidases, and the second one is endopeptidases. Okay, example for exopeptidases, we have your amino, so meaning the terminal amino group, and then carboxypeptidase, it degrades your terminal carboxylic acid group. Next, we have endopeptidases. Of course, this is the enzyme capable of attacking the peptide bonds within the polypeptide chain or your internal peptide bonds. So we have several examples. We have trypsin, chymotrypsin, pepsin, and papain. So here, class, we are going to focus on trypsin and chymotrypsin. Oh, both of them are in the carboxylic side of mechanism. Okay, again, ang mechanism nila is on the carboxylic acid side of your polypeptide chain. So for trypsin, it is only for lysine and arginine. O sir, familiar, parang basic amino acids sila both. Yes. So again, for trypsin, it can process the degradation of your basic amino acids. All right? Si chymotrypsin, o sir, familiar din. Those are all aromatic amino acids. Okay? So this is a certain or simple reaction mechanism for the mechanism of trypsin. O class, check nyo nasa carboxylic acid side, nasa carboxylic acid side siya ng lysine or arginine. So it's applicable to any amino acid except protein in the N-terminal side. Alright, so ayan, meron tayong did you know segment for your proteases. So first one, extremophile enzymes exist. So class, I think you are already familiar with a very known or prominent example of an extremophile enzyme. Diba? We use it in your PCR, your polymerase chain reaction. So some microorganisms called archaea live in environments that would be deadly to most life. So for instance, class, yung Taq polymerase. Diba? So this stuff, microbes produce extremophile enzymes that remain active under extreme heat, acidity, salinity, pH, or cold. Okay? So that's your extremophile enzymes. Ay, sir, meron ka pang did you know? Yes. Pineapples and itiba. O familiar with the enzymes that is present in your pineapple? So we call them your bromelain, diba? And that bromelain is a protease enzyme that can break down protein that is found in the surface of our tongue. Kaya when we are eating pineapples, you can feel some small tingling or your burning sensation in your tongue. So actually, that's your bromelain breaking down your surface of tongues. The surface proteins of your tongue. O, diba? So meron palang connection ang biochem with our everyday life. Okay, class, now that we are done with the introduction to enzymes, let's proceed with the unique properties of your enzymes. So of course, as I have already reiterated a lot of times, for catalysis ang main function ng enzyme natin. But of course, more than that, it has several property pa. Okay? So here you can see the GIF of your uncatalyzed versus catalyzed reaction. As you can see, kapag uncatalyzed, ang large ng activation energy. And for catalyzed, of course, it provides an alternative pathway that has lower activation energy. And of course, ang result, it enhances the reaction rate. Next, ayan, we have specificity. I think you are already familiar with this. Enzymes, again, are very selective or specific to the reactions or to the substrates they react upon, okay, or they catalyze. Then third, we have sensitivity, okay? Again, in general, ang proteins po, they are sensitive to environmental conditions fluctuations, all right? So, pwede silang ma-denature once they are not in the optimal conditions. And lastly, we can also regulate your enzymes, di ba? So, of course, hindi lang enzyme ang pwede mag-regulate. They are also regulated. All right, so class, in order for me to introduce to you the substrate binding, so let's watch a short video about substrate binding. So again, this is your intro to substrate binding.
Most chemical reactions do not occur spontaneously in a cell. Instead, cells rely on proteins called enzymes to kickstart chemical reactions and speed them up, enabling cells to get the most out of the energy sources available to them. Enzymes have a unique way of kickstarting reactions. They work by binding to one or more specific molecules called reactants, or substrates. Binding occurs at a special region on the enzyme called the active site. Once the substrates bind to the active site, they form what's called an enzyme-substrate complex. As the enzyme and substrates begin to react, some of the chemical bonds in the substrates begin to weaken, causing them to link together. Eventually, the chemical reactions at the active site lead to the formation of a different molecule. This is referred to as the product. Once the reaction has occurred, the product is released from the active site. The enzyme returns to its original state and is free to react again with another set of substrates.
All right, so that's our introduction to substrate binding. Okay, so class, from the video, this is the summary of what's happening under substrate binding. So again, class, recall, ang enzymes, they have an active site, all right, or they have a catalytic site. So here is the cyclic mechanism of substrate binding. In your manual, I guess it's in linear form. So similar lang siya, class. So here, our first step is the binding of your substrates to the active site. All right, and then of course, in the second position, second step, we will form the ES, or the enzyme substrate complex, which are held by weak interactions. It can be electrostatic, hydrophobic, or hydrogen bonding. All right. So upon binding, of course, your enzyme will find an alternative pathway in order to lower the activation energy and hence enhance the reaction rate. And after some time, of course, we will now your substrates or your initial reactants are now converted to products and released thereafter. And here you can see that the enzyme is already or again available for the next round of substrate binding. And as you can see, diba, your enzyme retain its structure. So, sir, for substrate binding, I think we have two different models. That's right, because here we have your lock and key and induced fit model. So actually, class, si lock and key model ang pioneering model for substrate binding. So very easy ang definition ng lock and key model. So it just suggests that your substrate and your enzyme are in perfect fit. Okay, so again, they possess specific complementary geometric shapes that fit exactly or perfectly into the enzyme. So do you think, guys, is this a valid reason? So somehow during their time, this is valid, that enzyme is rigid and unchanging during substrate binding. However, because of different experimentations, scientists have found that there is a transition state stabilization wherein this model cannot explain. That's why the induced fit model was suggested. Okay, again, induced fit model suggests that active site molds around the substrate upon binding based on dynamic recognition and interaction. So as you can see, class, here, there is a conformational change happening upon binding of your substrate. So that is more true compared to your lock and key model. So here, natitake into account yung na-observe na transition state stabilization through conformational change and with binding interaction. Again, guys, ang main clue dito is that lock and key model, rigid si enzyme. Okay? Sa induced fit model, dynamic si enzyme, meaning it undergoes conformational changes. Okay, so now let's proceed with enzyme kinetics. All right, class. So for enzyme kinetics, don't be overwhelmed because I guess this is the main point of confusion, okay, of the students. Here we are just going to focus on two parameters. So relatively easy naman ang nag-appear sa board exam. So from your manual, you can see there the two types of equation, si Michaelis-Menten and si Line-Weaver-Berg equation. So si Line-Weaver-Berg equation, class, this is just from your Michaelis-Menten equation by taking the double reciprocal of your original MME or Michaelis-Menten equation. All right. So the two parameters that I am pertaining to are your first one, ayan, si Michaelis constant. Again, si Michaelis constant or denoted as Km, this is your substrate concentration at half Vmax. All right? And of course, this is the summary of how are we going to interpret if it's a low Km or high Km. Pag low Km, meaning si enzyme may high affinity sa substrate. All right? And si high Km, meaning low affinity naman. So as you can see, they have an inverse relationship. And then the second parameter is your maximum velocity or the maximum reaction rate when the enzyme is saturated with substrate. All right? So this is a very important, class, because we will apply the kinetics, these two parameters, in finding or in determining the type of inhibition in your enzymes. All right? And that is our next topic. For enzymes, we can categorize them in two types, reversible and irreversible. All right? So reversible, class, we can further subdivide it into four categories: competitive, uncompetitive, non-competitive, or mixed. I think you are familiar with this four already, di ba? So of course, again, gagamitin natin ang parameters na Km and Vmax in order to determine what type of reversible inhibition. O, di ba? Sa irreversible naman, ayan, covalent po ang bonding ng inhibitor to the enzyme. That's why it permanently blocks the active site. So meaning very toxic po ang irreversible inhibition. Examples, we have cyanide. O, di ba? Familiar with cyanide poisoning? Carbon monoxide, rotenone, and antimycin A. So class, when we reach the electron transport chain discussion, these are actually the inhibitor of your ETC complexes. All right? So class, this is just an analogy of your reversible and irreversible inhibition. So irreversible meaning permanent ang damage. So reversible, you can reverse the inhibition. For reversible inhibition, we have four types. So again, first one is competitive. Ang main property ng competitive inhibition is that there is only one active site for substrate and inhibitor. That's why we call them competitive. Okay? For uncompetitive, here is the visualization. So as you can see, that's why it's called uncompetitive, because there is a separate site of binding for your inhibitor. But ang case here is that your substrate must be bound first to your enzyme before your inhibitor bind to its binding site. Sorry. So that's the characteristic of uncompetitive inhibition. All right? For non-competitive and mixed inhibition, they are actually the same lang. Ang difference lang nila is yung affinity between the E and ES complex. As you can see, class, both of them, they have two binding site. One is the active site and one is the inhibitor binding site. So of course, if you will ask, sir, ano po ang difference between the two? So si non-competitive, it has an equal affinity with your enzyme or ES complex. Okay? Pwede mag-bind sa inhibitor kahit wala pa si substrate, or pwede rin siya mag-bind pag nandun na si substrate sa active site. Si mixed inhibition, it has an equal, okay? Hindi pantay na affinity with your enzyme and enzyme substrate complex. All right, okay? Sir, pwede pong pa-summarize ng reversible inhibition. Okay, so I think this is also in your manual. Ayan, so class, this is the table that you need to familiarize. Sir, parang ang hirap niya. Baka naman may mnemonics ka. Okay, so of course, class, this is actually challenging for students to memorize. So I will give you a mnemonic that can help you in familiarizing this easily. So again, we have four inhibition types, and this is the mnemonics: cis, mid, normal safe delivery, and up, dharma, down. Okay, so I will explain how to use this mnemonics. First, cis. So these three letters corresponds to your mechanism of action ng competitive inhibitor. C meaning competitive, I meaning increase, Km. So paano namin malalaman which is which? So of course, class, yung Km yung second letter, si Vmax yung third letter, sorry, alphabetical lang. So meaning cis, competitive for C, I, increase, S meaning same or no effect kay Vmax. All right? For mid, ayan, M for mixed, I, increase Km, D, decrease Vmax. Thirdly, we have normal safe delivery, N meaning non-competitive, S meaning same or no effect, D meaning decrease, and lastly, ayan, up, dharma, down. Uncompetitive will decrease both Km and Vmax. So I hope that's clear, class. So you can use the mnemonics in order to familiarize yourselves easily with reversible inhibition types. But for now, I will proceed with a supplementary discussion on another type of inhibition. So we already have your irreversible and reversible. So aside from the four reversible inhibition, we also have your feedback inhibition. Okay? So feedback inhibition is where the end product of your pathway inhibits the first enzyme of that pathway. So sometimes ang main mechanism ng feedback inhibition or your regulatory enzymes ay through allosteric modulation or reversible covalent modification. So again, class, I would like to emphasize, so feedback inhibition is a type of reversible inhibition. All right? In order to visualize, here is the process of feedback inhibition. As you can see enzyme 4, it has a product that will inhibit the activity of enzyme 1 in that same pathway. All right, and then lastly, second to the last, we have enzyme activity. Of course, as we have discussed earlier, very sensitive C enzyme, and its activity is influenced by several factors. So I think we have 4 factors here: substrate concentration, enzyme concentration, pH, and temperature. Okay, let's discuss this one by one. For substrate concentration, as you increase the substrate, mag-i-increase din C activity, provided that your substrate concentration is enough. All right, it's not limiting. For enzyme concentration, parehas lang po. So increase enzyme concentration will increase activity, until it becomes saturated. Ayan. Thirdly, pH. Ito class very crucial, particularly sa biological systems. So each enzyme has an optimal pH. And of course, if you deviate from their optimal pH, it can denature them, making them inactive, or it can lower their activity. And lastly, temperature. Oh, sir, kala ko ang temperature may optimal temperature din for each enzyme. No. So generally, ang trend for temperature is that as you increase the temperature, it will also increase the activity up to a point. Alright? So somehow, meron pa rin palang optimal range of temp for your enzyme, after which your protein denaturation will occur, thereby inactivating your enzyme. Okay? For the mnemonics, ayan, your favorite part, we have PETSA, pH, enzyme concentration, temp, substrate concentration for activity. Ayan. And for our supplementary discussion, so class supplementary na lang to ha. So we also regulate your enzyme. So we know that enzyme regulates reaction. But of course, we also need to regulate your enzyme in order to control their concentration in your metabolic pathways. So very crucial in maintaining homeostasis. So we have 4 techniques in regulating your enzymes. First one is allosteric regulation. So this one, you can compare it to your switch on and switch off. All right? So we have a regulatory molecule that can bind to an allosteric site in an enzyme that will alter its activity. For example, reversible inhibition. The 4 types, including feedback inhibition. Next, we have covalent modification. Ito class, very known example is phosphorylation. All right? But still reversible pa rin, even though it's covalent modification of your enzyme. Thirdly, we have proteolytic activation. So this is the main regulation for our zymogens. Familiar with zymogens? These are your inactive enzyme precursor. So using proteolytic activation, we can activate your zymogens, for instance, trypsinogen to trypsin via cleavage of a repressing unit. And lastly, protein degradation. So complete degradation to meaning irreversible. Okay? So they are degraded when they are no longer needed to control the metabolic flux. Alright, so class, to end our discussion on proteins, amino acids, and enzymes, here are 2 sample problems. Okay, please try to answer this before checking the answer in your manuals. First, PMSF inactivates serine proteases by binding covalently to the catalytic serine residue at the active site. So this enzyme inhibitor bond is not cleaved by the enzyme. What type of inhibitor is PMSF? Sir, alam ko yung clue dyan. Yes, okay class. Ang clue natin here is covalent bonding. O diba, as we have discussed earlier, this covalent bonding in terms of inhibition is a characteristic property of an irreversible inhibitor. So this is the structure of your PMSF. As you can see, slightly similar kasi ang structure niya with the substrate of your serine protease. That's why it was able to inhibit through irreversible inhibition. Okay? Number 2, an unknown molecule is added to an enzyme catalyzed reaction, immediately decreasing its rate. If the addition of more substrates has no effect, but the addition of an antibody for the unknown molecule restores the initial reaction rate, what type of inhibition is most likely occurring? Okay, so let's spot first the clue in your given. So here we have a determining hint na decreasing rate. So meaning in terms of our parameter, decrease daw si Vmax. Okay, so decrease the Vmax, and then again, if the addition of more substrates has no effect, so class, when we are talking about substrates, it has has something to do with your Km or your Michaelis-Menten constant, okay? So of course, if no effect meaning same po si Michaelis-Menten. Same Km, decrease Vmax, okay? So that is a characteristic of your, what do you think class? So use the mnemonics, diba? Normal safe delivery. Non-competitive, same Km, decrease Vmax. Diba? So our answer here is your inhibition here is non-competitive. Okay, so that's it for our discussion on proteins. So class, please answer PT3 10 items for 15 minutes. So use your PT booklets, and then of course from your preceding practice tests, please put your scores in your PT booklets, in front of your PT booklets. Okay? So our next topic is nucleic acids. Alright, so class, this is the last topic on our discussion on biomolecules. So I hope you are ready and excited to finish our first batch of discussion for our biomolecules. So in particular, we are going to focus on